Hello, everyone. Welcome to Unit 4, Day 4, and today we're going to continue our discussion of transport and specifically diffusion. So we're going to be talking about passive transport, and we're going to be doing some practice problems with diffusion. So let's go ahead and get into it. All right, so today's aim is how do molecules move across a cell membrane in passive transport? So we know that passive transport is another word for diffusion at this point. We know this, right? And a couple of things we know about diffusion. Right, we know it occurs without energy. We know it occurs from an area of high concentration to low concentration, right down the concentration gradient. And these are the main bullet points, literally, about diffusion that we want to remember. Right, so right, so if this was like on your actual paper in class, you would like circle the word and write above it, but this is the word version of that. I'll make a different font to get chaotic with it. But right, so when we see diffusion, we want to remember that it is transport that occurs across a semi-permeable membrane without energy from an area of high concentration to low concentration, usually non-particles. Cell membrane. This is also how we get most of the energy from the food that we eat. All right, so the do now. Molecules A and B are both organic molecules found in many cells. When tested, it is found that molecule A cannot pass through a cell membrane, but molecule E easily passes through. Might remind you of an experiment we just did. State one way the two molecules could differ. That would account for the difference in the ability to pass through the cell membrane. Right, so we can see here, or no, sorry, that's another question. So obviously, similar to the diffusion through membrane, molecule A, starch molecule, too large to diffuse across the cell membrane, and molecule B be a monosaccharide, such as glucose, is small enough to pass through the semi-permeable membrane. And one could also be an ion, right? We just said a few things. There could also be a higher concentration of molecule B on the other side of, or excuse me, of molecule A on the other side of the cell membrane. So it could not be able to pass through because it would be going against the concentration gradient. There's a lot of different answers, but this is the simplest one. All right, part A, diffusion practice. What process accounts for the change shown in lab setup A? So let's take a look at the diagram. We have here a beaker, and in the beaker there is a sugar cube and some water, and it looks like time passes and the sugar molecules begin to diffuse into the water, and then over time the sugar molecules have become equally distributed, right? Equilibrium has been reached. And then in lab setup B, there is some sort of a membrane between the two, and it looks like that's labeled as structure Z. And the sugar molecules do not appear to be able to diffuse across structure Z. So what process accounts for the change shown in lab setup A? Diffusion, the sugar Q dissolved in the water, and the molecules of sugar over time diffused until they reach equilibrium within the beaker, and a sugar water solution was created. Right now, this might settle over time, depending on, the sugar, what is being used, or it might create a homogeneous solution. But at this point, right, this is now sugar water. This is a new solution. This is a chemical change. In lab setup B, structure Z prevents the movement of sugar molecules into side 1. What part of a living cell serves the same purpose as structure Z? Why, the cell membrane, of course. In lab setup B, the sugar molecule that is not able to diffuse through structure Z can likely be identified as which type of sugar molecule? Support your answer. So, due to its inability to pass through the cell membrane, I don't know what kind of like disaccharide or polysaccharide would be sold in a sugar cube, but due to the inability to pass through the cell membrane, the sugar molecule is most likely a disaccharide or polysaccharide whose molecules are too large to pass through without first being digested. Right, so they're like the starch molecules in diffusion through a membrane lab, they can't pass through the cell. Explain what could be done to the sugar molecule that would allow for diffusion to occur. So we need to break it down, right? How do we do that? By adding an enzyme to the solution, by heating the solution, or adding a pH solution, or manually stirring the mixture. So all of these things these actions would simulate digestion, which is what the human body does to allow polysaccharides, disaccharides to enter the cell. Right, so enzymes, heating the solution, all of these different things might work. But ultimately, what needs to be done, they need to be digested. They need to be broken down. All right, so now record your observations using the diagram below and be sure to be as detailed as possible. So remember I told you guys that you count the molecules? Well, that's what you're doing here. So you should pause the video right now and count and record your observations and then press play and go through it. Okay, so pause the video, count the molecules, and make your notes and then press play. Okay, hopefully you paused, but all right, so we can observe that there are 1, 2, 3, 4, 5, 6, 7, 8 water molecules inside the cell. Four. And then there are 1, 2, 3, 4, 5, 6 water molecules inside the cell after. So 2 water molecules diffused out is our, analysis. 6 water molecules inside after, right? So connecting this back to Unit 1, our observations are directly supported by our data. Our analysis or our inferences are the connections that we can make based upon what we see here in the diagram. So the oxygen molecules, there are 2 inside of the cell at the beginning and 1, 2, 3, 4, 5, 6 outside of the cell. So therefore we know that the oxygen is going to diffuse into the cell. Oxygen should diffuse into cell, and it does. We can see that right there is more oxygen inside the cell. Down concentration gradient. And that's also what we observe with the water. Protein molecules, too large, complex, 2 cross cell membrane. So now here you can count them out. 2 oxygen molecules inside cell 4. And this is connecting to our second bullet point of analysis. 4 oxygen molecules inside cell after. And 2 protein molecules inside cell before and after protein molecule. All right. I think that is enough for this table. If you observed anything else or made any other connections, that's super cool. But if you didn't, that's enough. Feel free to comment below if you made any connections that I didn't. But yeah, that's what we're doing there. And now we're on to the last question. So the diagram below shows the relative concentration of molecules inside and out of the cell. And you can see here, wow, we're counting little pictures again. So which statement best describes the general direction of diffusion across the membrane of this cell? So we can see here glucose would diffuse out of the cell. So this one is wrong. Protein would diffuse out of the cell. We know protein is too big, so that's wrong. Carbon dioxide would diffuse out of the cell. Well, let's see how many carbon dioxide we have inside and outside. It looks like inside the cell we have 1, 2, 3, 4, 5 carbon dioxide, and outside there's only 1 carbon dioxide. So the concentration of carbon dioxide is higher inside the cell than outside the cell. So it looks like carbon dioxide would diffuse out of the cell. Let's double check and make sure that's our answer. Oxygen would diffuse into the cell. It looks like there's oxygen, 1, 2 oxygen outside the cell, and 1, 2, 3, 4 oxygen inside the cell. So oxygen would also diffuse out of the cell. The concentration of oxygen is also higher inside the cell than outside the cell, so it wouldn't move against the concentration gradient. It would move down it or along it. So the correct answer for the last question, number 7, is C, and that's all I have for you today, ladies and gentlemen. Today's aim was how do molecules move across a cell membrane in passive transport, and we were talking all about diffusion. Don't forget to do your exit ticket, and please go ahead and comment on this video if you had any other observations that I didn't mark here or any other analysis or connections. Please also go ahead and like and subscribe to my channel so you can see when I post new videos almost every single day. And go ahead and hit that little bell so you get a notification every time I post a new video. Okay, that's enough for my YouTuber hyping. Please, ladies and gentlemen, make sure that you go ahead and submit that exit ticket and your guided notes. And I'll see you all tomorrow on Zoom. Bye, guys.